Okay, hello everybody, and welcome to today's Sunday webinar. Welcome to a few new people who've not joined me before. I'm glad you were able to join me. Hopefully you went to Socrative, hopefully you did the quiz beforehand. I know it's quite hard, and hopefully the answers would have come up automatically, so it would have stimulated a bit of thought about the topic of gas exchange. The next one is in 2 weeks. I'm taking a weekend off. It's in 2 weeks' time. I'm going to do genetic crosses, monohybrid, dihybrid, sex linkage, autosomal linkage, chi-squared, etc. So if you want to register for that, then you can. And sorry about the error in booking. I had put the wrong settings into Eventbrite. So those of you who were unable to book last week, hopefully everything is sorted now. Any point you can ask questions through the question and answer box at the top or in the Zoom chat. Or you can leave the questions till the end if you want, or you can ask questions as we go through. Okay, so let's make a start. So we're going to look at gas exchange surfaces, and we're going to talk about why you need gas exchange surfaces and gas exchange surfaces in humans, fish, and insects. Now, the need for a gas exchange surface results from the volume of an organism. So when you've got lots of layers of cells, it means that less oxygen can diffuse into the organism as it needs to be taken up by the cells that it's going to go through. The more active an organism is, the more oxygen it will need to get to its tissues. Because remember, the oxygen is being turned into water and the carbon dioxide is being produced through Krebs cycle and the link reaction in the mitochondria. So the organism needs to get in oxygen and needs to get out carbon dioxide. Now, if an animal is very active, so here we've got this leopard running around, it will need more oxygen. And if it's less active, like the sloth, it would need less. Remember, there's also a difference between if you're an ectotherm or an endotherm. So endotherms are going to require more oxygen because they have a temperature which is higher than the ambient temperature, whereas an ectotherm has temperature like the snake, which is the same as the ambient temperature, so that it won't require as much oxygen. Surface area to volume ratio. This is where people have to remember that cubes have 6 sides. As you increase the size of something, the amount of surface area that it has relative to its volume falls. So if you have a cube of side length 1, then it has a surface area of 6 and a volume of 1, so surface area to volume ratio of 6:1. If we double the size of the cube, the surface area to volume halves. If we treble it, it goes down 3 times to 2:1. And you can do a mental experiment thinking that if you took this block and you chopped it up, then thinking about how the surface area would change relative to the volume. Now, if you're a very small organism, you don't need a separate transport system. The cells are surrounded by the environment they live in, so they can just obtain the oxygen straight from the environment, and they have just diffusion alone. Because remember, gas exchange surfaces are all about enabling sufficient diffusion to occur to supply the needs of a large active organism. So you're doing the things to increase diffusion, or the cells have evolved, the organisms have evolved to have surfaces which increase diffusion. So, for instance, a very small organism like an amoeba is extremely small, so that the oxygen can just diffuse across its surface and the carbon dioxide can diffuse out. So they doesn't need a specialized surface for exchange. And when you get larger, as soon as you have an anatomy which is more than 2 cells thick, diffusion is too slow. So there needs to be an adaptation to improve gaseous exchange. So here is a flatworm. Now, as you may think, the flatworm's speciality is that it's flat. So this means it has a very large surface area compared to its volume. If you think about taking a piece of plasticine and hitting it with a hammer, then you won't change the volume, but you do change the surface area. So you've got your flatworm here. It's dorso-ventrally flattened. It can be several centimeters long, but it's extremely thin. So therefore, no cell is very far from the surface, and simple diffusion is enough. Now, as you get larger, for instance, an earthworm, you do need specialized surfaces and adaptations for exchange. So the earthworm is still exchanging oxygen and carbon dioxide across its skin. It's got a large surface area than it would do if it was just a spherical blob, because it's long and round, and it's also got folds on it which help to increase the surface area. But it also needs a transport system. So earthworms have a closed circulatory system. The problem is that the cells in the middle of the earthworm are a long way from the surface, so diffusion is not sufficient. So it's got hemoglobin, it's got a respiratory pigment which takes up the oxygen, then it's got a circulatory system to move that oxygen around the earthworm so they can get to cells deep in the body. And the outer layer is thin and moist so that the gases can diffuse across the membranes faster. Okay, so common features of gas exchange surfaces. Now, whenever you're talking about gas exchange surfaces, be they gill lamellae or alveoli or tracheoles, you've got to think they have a large surface area. It gives them a large surface area. They're permeable, they're thin, they're moist, and they have a mechanism to maintain the diffusion gradient. And all of these things are true about all of the gas exchange surfaces we're going to look at. So how do you achieve a large surface area relative to the volume? By folding the gas exchange surface and having lots of alveoli, lots of gill lamellae, lots of tracheoles. It needs to be permeable to allow diffusion of the gases. It needs to be thin, so you have a short distance for diffusion. So what's thin is the distance between the air and where it needs to get to. So the epithelial cells around the alveoli are very thin. The epithelial cells around the gill lamellae are very thin. So you've got a very short distance for diffusion. It needs to be moist, and it needs a mechanism to maintain the diffusion gradient. So on one side of the surface, you need a ventilation mechanism. Ventilation is movement of the respiratory medium over the respiratory surface. So the respiratory medium could be water or air. And on the other side, you're going to move the blood, so that maintains a concentration gradient. Okay, so we're going to have a look at some large multicellular organisms. Problem with a large multicellular organism, you've got a low surface area to volume ratio of the outside of the organism. Diffusion would be too slow and the distance would be too great. High metabolic rate, they need a lot of oxygen, therefore you need specialized surfaces for exchange. So fish have gills, and you can see the level of vascularization here because they're very red. We've cut away the opercular flap here in this fish. Insects have tracheae and tracheoles, and birds, reptiles and mammals, they have lungs. And here are some more exchange surfaces. So these are gills on the surface. I think this is some form of a sea slug, and this is here an axolotl with some external gills. Nice cheery chap. Amphibia, just to mention these, amphibia come up occasionally. So amphibia, remember, live in water or have to return to the water for fertilization because they have external fertilization, because they haven't evolved internal fertilization like other things that live on the land. The larvae that they have, the tadpoles, have gills, and then the adults, which live predominantly on the land, these, remember, are dorso-ventrally flattened. If you look at a frog from the end on, they're quite thin. So they've actually got a large surface area to volume ratio by being thin and flat. Plus they're ectotherms, plus they're not very active. They don't tend to go hopping past you as you go to college in the morning. So they obtain a lot of the oxygen across the skin unless they're very active, in which case they can get it from the lungs as they hop around. Okay, so let's have a look at fish. So fish have got an issue, mainly that they live in water, and there isn't much oxygen in water, and water is pretty hard to move. So fish have got a very specialized gas exchange surface, which is very clever. So the problems with water, it's got a very low partial pressure of oxygen, it's dense, and it's more viscous than air. It's harder to move. So the bony fish has got the opercular flaps, which are here and here. And this is where the water exits. So the water is going to enter through the buccal cavity and then exit out of the opercular flaps. Here we cut away one of the gill operculum flaps, and you can see the gill arch and then the gill filaments radiating away. So the gills are made of lots of folds, giving you a large surface area to maximize gas exchange, good blood supply, and a very thin layer of epithelial cells which are around the blood to separate the blood from the water. Here's a mackerel gill, this bit's known as the gill arch, and then we've got pairs of gill filaments coming away. Here's a cross-section through the gill. These are the surfaces of gas exchange. So these are the gill plates or gill lamellae. The two terms are interchangeable, but I'll use the term gill lamellae. And these are on the gill filaments. And because the fish has got a lot of gill lamellae, they have a large total surface area. Here is zooming into an individual gill lamellae. And what you can see, here's an epithelial cell. And here are some red blood cells. Now, what you can see here is there's an extremely short distance for diffusion of oxygen in and carbon dioxide out. Now, gas exchange in fish. We've got 4 pairs of gills in the pharynx. So there's 4 gills here. And the water flows in through the mouth and out over the gills. Now this is a one-way flow system as opposed to the tidal flow that you have in humans. Now the clever thing in bony fish is that the deoxygenated blood enters and goes the opposite direction to the water. This is known as countercurrent flow. And this is an extremely clever adaptation that we're going to talk about quite a lot. So water again flows in, over the gill arches, goes through the gill filaments, and here going over the gill lamellae in the opposite direction to the flow of blood. More pictures, fairly obvious, talked about that. Now, countercurrent flow. So let's have a chat about this with the whiteboard. So countercurrent flow is the fact that the water flows in the opposite direction to the gills, to the blood in the gills. Now, why this is important is it enables the blood to leave the gills being far more saturated than it would be if the blood was flowing in the same direction as the water. Now, the way I try and explain this is to imagine you had 2 pipes. So you had a hot pipe and you had a cold pipe, and they're running next to each other. And the hot pipe is at 100 degrees and the cold pipe is at 0 degrees or 1 degree. Let's say it's liquid. Now they're flowing alongside each other. So if we think that the pipe is infinitely long, then the hot water is going to get cooler and the cold water is going to get hotter. And at the end of the pipe, they will have reached an equilibrium of 50 degrees and 50 degrees each. Now, if we plotted this on a graph, where along the bottom we had distance along the pipe, then we would see that the hot water would get cooler and the cold water would get hotter until they reached 50 degrees. Now, Imagine if we reverse the direction. So instead of them going in the same direction, they go in the opposite direction. So you have water entering at 0 degrees and hot water entering at 100 degrees. The hot water will always be encountering cooler water, and the cold water will always be encountering hotter water. So at the end of the tube, the cold water would be at 100 degrees and the hot water would be at 0 degrees. Because even if you think about the middle of the tube, the 50-degree water in the hot water is going to be encountering 45-degree water in the cold. So the 50 will be cooling to 45, but it then encountering cooler water, which is at 40. So there's always a gradient of temperature. You're always losing heat. So if we plotted this on a graph, we would see that the cold water, which started off down here, will be getting hotter. And the hot water will be getting cooler. Now, if we take that metaphor and then we say, well, let's think about it in terms of oxygenated blood. Hopefully you can see that if we replace temperature with oxygen, then if we had oxygen in the water, and say put 10 here, so 0 oxygen, actually get that wrong, so try that one again. You've got 0 oxygen in the blood and say 10 oxygens in the water, then if they run alongside each other, then they'll reach 5 and 5. So that the saturation of the blood will be at an equilibrium with the saturation of oxygen in the water. But if we run them in an opposite direction, then the blood is going to exit the gill far more saturated than it would do, because the oxygen in the blood will go up and up and up and up, because it's always encountering more oxygenated water. So the advantage to the fish of this system is that its blood is far more saturated with oxygen because of countercurrent flow than it would be if it was parallel flow. So this is a tremendous advantage and a tremendous piece of evolution. Okay, back to the slideshow. I hope that made sense to people. So, countercurrent flow, blood in the capillaries flows in an opposite direction to the water. The dissolved gases diffuse faster between fluids with a large difference than with a small difference. However, what's going to happen here is that the oxygen is constantly going to be diffusing out of the water into the blood, so that the blood will come out of the gill lamellae far more saturated than it would do with parallel flow, because the concentration gradient is maintained over the entire distance of the gill lamellae. That's all I talked through. Yeah, the advantage to the fish is that the blood is more saturated with oxygen than it would be with parallel flow. So breathing in fish, or ventilation in fish. So the way that they do this is they alter the volume of the chambers. So they alter the volume of the buccal cavity, which is the mouth, and they alter the volume of the opercular cavity, which is behind the gills. And by altering those volumes, they cause water to flow over the gills. So the first step is take in water. First step is open the mouth and then close the opercular flaps at the back. Lower the floor of the mouth That increases the volume of the mouth and the pressure falls so that water moves in. Then close the mouth, raise the floor of the buccal cavity, and then open the opercular flaps, and then that will cause water to flow from a higher pressure in the buccal cavity to a lower pressure in the opercular cavity, which will cause the water to flow over the gills. So, breathing in, open the mouth, expand the volume of the buccal cavity, close the opercular flaps. And then breathing out effectively is close the mouth, contract the buccal cavity, move the opercular flaps out, and that causes the water to flow over the gill filaments. Okay, gas exchange on land. So the problem with being on land is you've got evaporation of water from the surface of the lungs, and plus you've got the loss of heat if you're an endotherm. So internal lungs minimize the loss of water and try to minimize the loss of heat. Lungs are in amphibia, reptiles and birds, and insects have got a completely different system, that they have a tracheal system. So you need to conserve water. The gas exchange organs are inside the body rather than having external gills, and that helps you also to reduce the loss of heat. So let's have a look at humans. So you need to know the gross anatomy of the gas exchange system. So first of all, you know that the lungs are in the thorax, hopefully, and they're in an airtight compartment, which has got the pleural membranes and the ribs and the diaphragm around the outside. Now remember, the lungs, the lung tissue is extremely delicate. You can't attach muscles to it and start pulling it around. So the way you change the volume of the lungs is by altering the volume of the box that they're in. So the lungs are in a box which is only got one entrance, which is the trachea, which then splits into the two bronchi. Around the outside of the box are the pleural membranes, and then you've got the ribs and the diaphragm. And the action of the ribs and the diaphragm is to change the volume of the box. When you change the volume of the box, you alter the pressure that's in the box so that air then moves in or out. So only one entrance via the trachea. Now, let's have a look at the trachea. So first of all, you've got a larynx at the top. The trachea then runs down from the larynx to the carina, where it then splits into the two bronchi. The bronchi then split into bronchioles. Looking at a cross-section through the trachea, you've got a C-shaped ring of cartilage. And the reason it's C-shaped is because behind the trachea, you've got the esophagus, down which you swallow lumps of food. If it was a solid ring of cartilage, it would make swallowing food considerably more of a challenge. Also down the trachea, you've got ciliated epithelium and you've got goblet cells. And the purpose of those is to keep your lungs free of dust and pathogenic bacteria. You've also got smooth muscle, and smooth muscle can alter the diameter of the airways. Remember, the smooth muscle is not under conscious control. It's under the control of the autonomic nervous system, not the somatic nervous system. Okay, more lunginess here. So what have we got? We've got a trachea, we've got a bronchus, we've got ribs, and then we've got intercostal muscles. Remember, there are two intercostal muscles and they're at a different angle to each other. So when you contract the external intercostal muscles, the rib cage moves up and out. And when you contract the internal intercostal muscles, which are at a slightly different angle, the rib cage moves down and in. If you've ever eaten spare ribs in a restaurant, then you've been eating piggy intercostal muscles. Now, underneath the ribs, you've got a diaphragm. The diaphragm is a sheet of muscle. Remember that muscle can only contract, it can't expand with force. So the action of the diaphragm is to go from a domed shape to being a flattened shape, which increases the volume of the thorax and therefore would decrease the pressure, which would cause air to move in and therefore inflate the lungs. If you remember, I keep coming back to this, the whole concept is you're changing the volume of the box that the lungs are in. Once you split from bronchioles, smaller and smaller bronchioles, you get to the alveoli, and the alveoli are the site of gas exchange. So if you're ever asked in an exam, what's the site of gas exchange in humans? Don't write the lungs, write the alveoli. So having a look down through the bronchi and the trachea, remember you've got ciliated epithelium with cilia on the surface, and they beat to waft the mucus up to the esophagus, where you then swallow it. And the mucus is produced by the goblet cells. So goblet cells secrete mucus. The reason they're called goblet cells is they're meant to look like a wine glass or drinking goblet. And they produce a glycoprotein, which is the mucus, which traps the any bacteria or dust. And then the cilia help to waft them up to the esophagus. You then swallow it and kill it in the stomach acid. So C-shaped rings of cartilage. These are a bit like the rings on a Hoover hose because they enable flexibility, but they also stop the sides of the trachea and the bronchi from collapsing in under the negative pressure when you breathe in. So the reason why these rings of cartilage are to prevent the walls of the trachea from collapsing inwards, which is what they'd do when you breathe in if you didn't have them. Smooth muscle that's in the trachea and the bronchi. Remember, the purpose of that is that it can constrict, which would reduce the diameter of the lumen, reduce the diameter of the bronchi and the bronchioles, and therefore restricting the flow of air to and from the lungs. And also then reducing the rate of water loss and reducing the rate of heat loss. You've also got elastic fibers in the alveoli. Now, when you inhale, the alveoli increase in size, which would increase the surface area for diffusion. So the alveoli expand as the air enters. And then when you stretch those elastic fibers, when you then relax the diaphragm or relax the external intercostal muscles, then the fibers then recoil, which helps push more air out. So they expand when you inhale, and then when you exhale, they recoil back, pushing air out. Alveoli are well ventilated, and ventilation, remember, maintains a diffusion gradient. They've got a good blood supply because they have an extensive network of capillaries. Now, if you're asked about adaptations, an adaptation is not that it has a good blood supply. The good blood supply is the consequence of having lots and lots of capillaries. So take care when you're phrasing that sort of question, phrasing that sort of answer, that you say what the adaptation is and then what the consequence of that adaptation is. So the adaptation, it has a lot of capillaries around the outside, and the consequence is it gives it a good blood supply, which maintains the concentration gradient. So by ventilation and the capillaries, you're always taking away oxygen and you're always bringing carbon dioxide. Very large surface area through having lots of alveoli. Do make sure you say lots of alveoli in any exam answer, because alveoli don't give it a large surface area to the lungs. Having lots of alveoli does. They're thin. The epithelial cells are only 1 cell thick. So here is an epithelial cell, and that's one thin cell so that oxygen can diffuse across a very short distance and then across the epithelium of the capillary and then into the red blood cell. So it maintains a very short diffusion pathway. Remember, the reason why you breathe and the reason why you pump the blood around the alveoli is to maintain a diffusion gradient. So the oxygen is diffusing into the capillaries and the carbon dioxide is diffusing out. A few other things. You've got pleural membranes around the outside which have pleural fluid, and that's a lubricant, prevents friction between the thorax and the lungs. And then you've got a surfactant, which is a bit like sort of a soapy washing up liquid. That's on the surface of the alveoli, and it prevents them from collapsing by reducing the surface tension of the water, because remember, the alveoli are moist. Okay, on to ventilation. So ventilation means you're supplying fresh air to replace used air. Movement of the respiratory medium over the respiratory surface means that there's always a higher concentration of oxygen in the lungs than in the blood, and that the concentration of carbon dioxide in the lungs is lower than in the blood. So this is what happens in inhalation. So inhalation is an active process. It requires energy. Most of the time, exhalation is a passive process that doesn't require energy. So inhalation is caused by the contraction of the diaphragm, which increases the volume of the thorax. Therefore, the pressure decreases, therefore air moves in. It's also achieved by the movement of the external intercostal muscles, moving the rib cage up and out, which also increases the volume. Exhalation is achieved by relaxing the diaphragm, and then the gut that you've pushed down when you breathed in is going to push back up again, which is helping to push air out. Also, you've stretched the elastic fibers in the alveoli so that that helps to push air out as well. When you relax the external intercostal muscles, which are between the ribs, the weight of the rib cage will push down on the lungs, and that will help to push air out in exhalation. So in inspiration, we move the lungs up and out, and we move the diaphragm down, which means that air moves in. Combined effect is to increase the volume of the lungs, and air moves in. And then in expiration, passive process, you relax the diaphragm, it goes back to its dome shape. The liver and the gut help to push air out, and then you relax the intercostal muscles and the rib cage falls and pushes down. So rib cage falls under its own weight, diaphragm relaxes, and the stretched lung tissue helps to push air out. Decrease the volume of the thorax, increase the pressure, so air leaves. Here we go again. Diaphragm, talk to all of that. I'll stop. Right, forced expiration. Forced expiration, if you're singing or playing a musical instrument or playing the trumpet, then you contract your abdominal muscles, and that helps to push the gut up into your thorax, which helps to push air out. And you also contract the internal intercostal muscles, which pushes the rib cage down and in. So the internal intercostal muscles contract, pulling the rib cage down and in. Abdominal muscles contract, pushing the diaphragm up because it's pushing the guts up as well. Decrease the volume of the thorax, increase the pressure, so air moves out. Now, measuring lung volumes and lung capacities is achieved by a spirometer, which these wonderful people from the 1970s appear to be using. Many of you will have used this in class, and it looks a bit like this. Now, be careful. If they give you a piece of apparatus like this in the exam, remember that all you're doing is you're breathing air that's in this box. When you breathe in, the air moves into the lungs of this chap here, so that the volume of the box will fall, so the pen will move down. So if you get a piece of apparatus like this, remember that inhalation will cause the pen to move down. So breathe in, breathe out, breathe in, take the biggest breath in, followed by the biggest breath out, and the vital capacity would be there to there. Not very good drawing, but I've got a better one coming up in a minute. So be careful with this apparatus. Make sure that you're fully aware of what they're showing you. If they've got the trace on the chymograph, it may be the opposite to what you expect. When you breathe out, it goes through a carbon dioxide absorber. This means that the carbon dioxide you breathe out will be absorbed. So any change in volume of this box is purely due to the absorption of oxygen by the subject. And the oxygen, remember, is being turned into water in aerobic respiration, because oxygen is the final electron acceptor in the electron transport chain in mitochondria. The subject is breathing out carbon dioxide, and the carbon dioxide is being absorbed. So any change in volume of the box is purely due to the absorption of oxygen. So the gradient of the line, if it goes down, that gradient will tell you the consumption of oxygen. Now, the speed of rotation of the chymograph, will give you time. So if they tell you it rotates at 2.5 millimeters per second, then you know that if you have 50 millimeters, that represents 20 seconds, and 150 millimeters would be a minute. So this was my word of warning about the chymograph trace. Be careful. It may well be the opposite from what you expect. What you expect is this, which is lung volumes, but if they give you a chymograph trace where they say on this axis it's volume of the chymograph, or volume of the spirometer, volume of the spirometer, not volume of the chymograph, so volume of the spirometer, then it's the opposite from what you'd expect. But that's increasingly rare because they realized that that was pretty mean to put that into exam questions. So this is what you normally get. You normally get tidal volume doing this. So tidal volume is between about 0.3 and 0.7 of a litre. The biggest breath in after the tidal volume is the inspiratory reserve volume. Biggest breath out after that is the expiratory reserve volume. The inspiratory capacity is the tidal volume plus the inspiratory reserve volume, and inspiratory capacity plus expiratory reserve volume gives you vital capacity. And the total lung volume is vital capacity plus residual volume. Residual volume is everything that's left in the lungs after you've taken the biggest breath out that you can. This is the point I was making about the gradient of the line. Remember that the fall in volume is going to be the result of oxygen being absorbed and turned into water, and then the carbon dioxide that's produced is going to be absorbed by the soda lime or potassium hydroxide or whatever it is that's absorbing the carbon dioxide, so that the fall or the gradient is going to give you the rate of oxygen consumption, because the distance it's moving along is time, and the fall in volume is purely due to the consumption of oxygen. Okay, on to insects. Now, insects we regard as being weird, but remember, most animals on Earth are insects. And we're the weird ones because we've got this bizarre lung and red blood cell and hemoglobin gas exchange system. Whereas insects, they went down a completely different pathway, is that they've got tubes running into all of their cells from the outside. So they've got a spiracle, and then they've got tubes, and the tubes split and split and split into smaller and smaller and smaller and smaller and smaller tubes. And these tubes go into almost every cell in the body. Now, insects often live in very arid environments, so they've got spiracles on the outside of the tracheae, and the spiracles open and close like valves, which helps to reduce the loss of water, because at the end of each tracheole there is some water. So because you've got a huge number of tracheoles, you then have a very large surface area through which you would lose water, so that the spiracles help prevent that loss of water. Now, the air sacs that you see in a large insect, these are definitely not exchange surfaces. They're not lungs. They don't contain alveoli. The air sacs are bellows to help push air through the insect to ventilate it. The gas exchange occurs at the tracheoles, so the ends of the tracheae where the oxygen diffuses into the cells of the insect. Now, an inactive or very small insect, diffusion is enough. But when you have a very large insect or a flying insect, the movement of the abdomen in and out helps to ventilate the tracheoles. A few more pictures. So we've got spiracles in each segment of the insect. Remember, the spiracles are valves over the ends of the tracheae. And then they split and 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 split to give you the tracheoles at the end, which have got fluid in them. So spiracles are opening, and then they go into chitin-lined tubes. Remember, chitin is a modified polysaccharide with a bit of nitrogen in it. It's a bit like cellulose, but it's a bit stronger. Smallest tubes are called tracheoles. At the end of it, you've got some fluid in which the oxygen dissolves and the carbon dioxide from the insect's tissues dissolves as well. Now, I like this diagram because it shows you've got a spiracle and then you've got these chitin rings, and the chitin rings help to keep the tubes open. Then at the end of it, you've got fluid, and the fluid is what the oxygen dissolves in and the carbon dioxide dissolves in. But look at this picture here. Look how small these tracheoles are when you compare them to a mitochondria. So these are tracheoles, which are individual tubes going into each of the cells from the outside. They're an amazing system. Now, this appeared in a recent OCR paper. I think it's the AS June one, the depth in biology. Was it? Yeah, depth in biology. And these are little mites. These are mites living within the tracheae, and they help suck this parasitic on the insect. And one of the questions was, how does this affect the insect? Well, it's going to affect the ventilation because it's going to cause more resistance to flow. But weird, aren't they? Okay, so the tracheal system gives them a large surface area because they've got a lot of tracheoles. Small insects have diffusion alone. Larger insects can ventilate the tracheal system by changing the volume of the abdomen and the thorax. So here we've got not a very good picture, but trying to get the message across of the ventilation of the thorax, and here are some air spaces changing in volume within a big insect. So the way they do this is that they change when the thoracic spiracles and abdominal spiracles are open. So if you had an insect, actually I'll do it on a whiteboard. So if we have our insect, and it's got thoracic spiracles and abdominal spiracles. So if it closes the thoracic spiracles, sorry, closes the abdominal spiracles, get my words mixed up, And then it opens the valves on the thoracic spiracles. And then if it increases the volume of the abdomen, then air will move in through the thoracic spiracles. Then it closes the thoracic spiracles. It opens the abdominal spiracles and moves the abdomen up to its original position, contracts the abdomen, and that will push air through and out through the abdominal spiracles. And remember, the purpose of the spiracles is to reduce the rate of water loss from the tracheoles. Okay, so back to this, we can see that we open the thoracic spiracles and at the same time you expand the abdomen. Then you close the thoracic spiracles and contract the abdomen at the same time as opening the abdominal spiracles. And that causes the air to be ventilated through the insect. Remember, this only occurs in active insects which are flying around. If you're small and not very active, then diffusion is fine. Now, the fluid in the end of the tracheole, when the insect is flying around, that fluid gets drawn up into the tissue, which achieves two things. So firstly, it supplies the tissue with oxygen because there's oxygen in that fluid, but also it's going to increase the surface area of the end of the tracheole, therefore giving more surface area for exchange. And when it rests, when the muscle's at rest, that fluid goes back into the tracheole and more of the carbon dioxide then diffuses out to the trachea. So during exercise, there's less fluid, so that increases the surface area of the end of the tracheoles. But after exercise, at rest, the fluid fills up. And then obviously the carbon dioxide diffuses out and the oxygen diffuses in. And then exercises is going to go back again. Okay, that's the end. If anybody has any questions, then now is the time to ask them. I hope you found it reasonably helpful.